Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Stuart McNall from KBI Biopharma.
Thank you, Leah, and thank you to everyone who's joined our webinar today. I'm Stuart, and I'm here to tell you how KBI Biopharma and Selective work together for cell line, process, and analytical development to speed clinical supply of emerging therapy products. First, a bit about the two companies, KBI Biopharma and Selective. We've actually worked together for the last 5 to 7 years, with Selective making best-in-class cell line development technologies that KBI has taken into our development labs to develop clinical processes for manufacturing to supply first-in-human product to support INDs and clinical trials. So our two companies actually, as of last year, are now under the same parent organization, JSR Corporation, based in Tokyo, with life science headquarters in Sunnyvale, California. Selective is, from the map, obviously based in Geneva, Switzerland, where it was founded, and they've developed quite a track record for taking cell lines to the clinic, which I'll show some data in a couple of slides. KBI is founded and headquartered in Durham, North Carolina, and that houses our mammalian cell culture manufacturing facility for clinical and commercial supply. And also the largest unit of our analytical services business was founded and is in Durham, North Carolina. Half an hour down the road in RTP, North Carolina, are the process and analytical development labs that are almost 100 scientists strong. And then we have microbial manufacturing and development in Boulder, Colorado. And then as of last year, our foray into cell therapy with a site in the Woodlands, Texas. So it's a very exciting field. Patients are being cured of rare diseases, so that's an exciting topic for another day. We also have analytical services offering in San Diego, California, when we acquired Alliance Protein Labs last summer, and a recent acquisition of Elion Labs in Boulder, Colorado, with first-in-class analytical services. So KBI is founded on analytics, and we've reinforced that foundation recently. But also, I'll tell you today about our significant growth in the development and manufacturing space and how we work with Selective in that regard. A bit of corporate overview on Selective. Founded in 2001 in Geneva. Really offering best-in-class cell line development technologies with some unique, genetic elements and workflows to achieve high-producing, rapid development of stable research cell banks, particularly for difficult-to-express proteins. Their track record for taking cell lines to make products that are clinical drug candidates is up to 95 of those candidates produced to date, and this number is continually growing. It's quite an impressive track record, particularly over the last 5 years or so. So lots of preclinical products as expected, but also on the right-hand side, you can see a number of phase 2, phase 3, and even commercial marketed products. So a well-established platform for cell line development. Jumping to the KBI history a bit. KBI is a company that was actually founded in 1997 under a bioreactor technology that later we developed into a KSEP single-use centrifuge. It's no longer part of the business, but certainly something we're proud of. But as the CDMO business started in 2004 in Durham, North Carolina, initially with the analytical services business. And from that time, we've worked on over 220 molecules for product characterization, formulation development, stability testing, et cetera. Drug substance, drug product release, of course. And then in 2010 is when we really dove deep into mammalian process development and manufacturing, building out significant lab space and expertise and manufacturing space and expertise. So we now have 3 2,000-liter bioreactors in Durham, North Carolina, with 2 purification suites to allow us to make clinical and commercial product. Right now, that's up to about 50 batches a year. On the bottom left, you can see the growth of our development and manufacturing over the years. So, definitely on an exponential growth there. And as I mentioned, really top-notch talent in this regard. Each of the last 2 years, we supported 12 or more INDs, taking molecules through development into manufacture. Those were not all Celexis based, but many of them were. And we also do a lot of late-stage process characterization, process validation to commercialize, to move products towards commercialization, which we have our first mammalian product in PPQ this year. But also we do standalone process characterization, scale-down process validation, and have all the DOE methodologies, et cetera. And then our microbial site has been commercially inspected and awaiting approval of the first commercial product there. Okay, just to round out the KBI experience, worked with a number of molecule types over the years, lots of mAbs, of course, but then you see, more and more of the biospecifics, fusion proteins, vaccines showing up there. And on the next slide, a quick snapshot of the distribution of these new modalities, what emerging therapies are coming to our door, and we feel like that's a pretty good representation of the industry as a whole. So the pie chart on the left, the fonts are a little small, but the blue are mAbs. So in 2016, more than half of the development and manufacturing projects we did were monoclonal antibodies. If you look to the pie chart on the right for 2017, the blue is proportional to the other biospecific or FC fusion or vaccine, modalities. So we feel like this really shows a bit of the trend of the industry. So, our conclusions are that, the non-mAb platforms are more and more important, and KBI has actually excelled in developing these non-mAb platforms that provide, a framework to build upon, that can be customized for your molecule, but the foundation is there so that we're minimizing, some of the development path and also building on our experiences to tailor processes for our clients. So with that, it's kind of the intro and the background, and so we want to do a polling question, and then I want to talk to you about how Celexis and KBI work together for rapid development. So Leah, I didn't know if you wanted me to read this off or if you wanted to do the poll.
Oh, you can go ahead.
Okay. So from what I can see on the screen here, the question may be a little chopped down, but the question is, for first-in-human development programs, what are your expectations for the time from gene to GMP, from the time you transfect a cell line to the time you have drug substance in your hand? What are your expectations around that? Realistic, what would you like to see? Or what meets your goals for getting to work towards an IND filing? So if everyone could please click your responses, and it looks like we have some coming in. All right, we're up to 12 responses. Please keep them coming. And Leah, are people able to still respond as I move forward in the slides, or no?
No, once we move forward from the slide, they won't be able to respond. So go ahead and get your responses in there.
Okay.
Okay. I think.
All right. Thank you for your responses. We have 17 responses. That's very valuable, very helpful. And let's get to results. Okay, very interesting. So hopefully I phrased the question well for you guys. But for the time you transfect a cell line to the time you have drug substance in hand, it looks like the expectations. People see a lot of 12 to 14 months, and then 10 to 12, and even 8 to 10 at the lower percentages. So, I think that's in line with what we hear from clients in the industry, and it's valuable information. Okay, so to have that speed for development, what are the things that stand out as needed? So, what we've seen over the years, integration of product quality testing with candidate selection and clone selection, we see as very important. You want to make sure you get the right molecule attributes and clones that behave very well. Definitely, the more you can integrate the cell line with the upstream, with the downstream analytical development, the faster you can go. Platforms certainly help in this regard, and equipment platforms that are well aligned with each other. So just in the interest of time, to get into a bit of the meat of what we do, so I want to tell you about our Selexis KBI integrated workflows, and the acronym we've come up with internally is SKI program. So with the Winter Olympics at hand, this is kind of a fun topic for us, but this is what we call our integrated programs for, essentially gene to GMP. The schematic here shows the timeline for the various stages at Selexis for them to make pools, to make the first clones, to do super transfection, and then do cloning from there. So what we do at KBI is we take those first clones, typically the first 12 clones, we bring those into process development. We'll pull those clones to do a supply batch, and then we'll take those clones into reactors to build on the upstream platforms, start pre-formulation development, downstream development, analytical development, et cetera. So we jumpstart the development engine, and then as the final SEC2 clones are available from Selexis, we typically take a handful of those into some final reactor sets to choose the top clone, to get into a 200-liter scale-up run in the development labs, which is often used for tox by our clients, and then scale up for manufacturing. And so doing all this, it puts us in GMP manufacturing before 9 months, so that we're well on our way to releasing drug substance and moving forward to enable your INDs. So just in the interest of time, to show a little bit of data here, this is our integrated workflow. On the left-hand side are the Selexis activities I described, and it shows how they flow into upstream process development, downstream process development, analytical development, pre-formulation development, and it all comes together at the bottom to enable GMP manufacturing. We rarely do engineering runs these days. We leverage our platforms. We do a 200-liter tox batch to culminate development, and the scale down is very well established between that 200-liter and our 2,000 liters bioreactors, which we run anywhere from 500 liters to 2,000 liters for production. To show you just a little bit of data, a snapshot of data, KBI has worked with over 20 Selexis cell lines to date for a variety of products, mAbs, fusion proteins, bispecifics, and others. And the table here gives you a feel for the types of titers we see. What stands out to me is that even for fusion proteins and bispecifics, we're often in that 2 to 3 gram per liter range, with some data as high as 5 grams per liter, which for those modalities I think is quite impressive. For mAbs, we've been as high as 9, some data points at 9 or 10 grams per liter, but those processes that went to manufacturing, in that 7 to 8 grams per liter range. And the example shown here for molecule A is for phase 1, we had just a couple of months to develop a process to get to manufacturing. We took the Selexis cell line. After a couple of months of work in the lab, 4.5 grams per liter. Later, as this client achieved breakthrough status, we did optimization to get the titers up, 7 to 8 grams per liter. So it's a nice story there. One thing, with formalizing our partnership with Selexis, KBI and Selexis being under one parent organization and working very closely together, KBI really wanted to bring our analytical capabilities to the table. I mean, I actually worked at another CMO for 15 years, and the analytical capabilities of KBI are well renowned. So we've built out these panels for different molecule types. For our clients to come to us, this is a panel that can do a lot of things for your molecule type. And then we sit down and figure out which assays they need where in the process. Typically we pick a couple of assays to look at candidate selection at Selexis, and then a couple of assays to do the final clone selection from Selexis. And then as we get into development, bring in a broader panel as you get towards reference material characterization, et cetera. So we're quite proud of our analytical capabilities and foundation at KBI, and it's fun for us to apply these technologies earlier and earlier in the cell line development process. So with that, just a conclusion slide that we've got this Selexis track record of first-in-class cell line development technologies going into now 95 clinical candidates. Over the last 5 to 7 years, KBI has worked with 20 of those cell lines, getting titers at 2 to some data points at 10 grams per liter, and applying our analytical capabilities. But the main thing is putting all this together. We are on our way to providing a first-in-human development platform that will provide gene to GMP manufacturing, in 9 months for mAbs. So, we haven't done 6 of those programs in completion yet, but we're well on our way on a handful of programs, and we look forward to coming back next year and telling you how those went. So thank you very much for your time. I'm glad to take any questions you have.
Thanks, Stuart. So you can go ahead and type your questions into the chat window. And our first question is, what cell lines do you primarily use for your mammalian development and manufacturing programs?
Yeah, so most of what I described today is CHO cell lines, and the Selexis, the terminology is CHO-M. So most of what I described on the mammalian front is CHO manufacturing.
And what mammalian cell culture manufacturing capacity do you have for clinical and commercial supply?
Yeah, so with our 3 2,000-liter reactor lines in North Carolina, we can supply up to 50 batches a year. So we have one stream that's the single 2,000-liter reactor that's our clinical stream. That can produce about a third as many batches. And then the other stream has the 2 2,000-liter reactors, and that can do clinical or commercial. So we maintain our clinical capacity flexibility, but those streams are at the commercial standards of operation and qualification.
And are many of your clients planning to launch commercial product from single-use bioreactors?
Yes, that's the trend we're seeing. There's a lot of industry forums like the BPOG forums that I've been a part of, and even the big pharmas are now launching out of 2,000 liters, and as their molecules show, lots of promise. Our vision for expansion is to expand to a 6 by 2,000 liter facility. That could, for a high titer and high throughput downstream process, you can make 1,500 kigs of product a year. So it depends on the modality. If you need 3,000 kigs a year, then at that point, people are certainly considering stainless. But yeah, many people are launching out of single use. They're certainly robust enough to do it. We're doing process performance campaigns this year in single use, and particularly with the new modalities. If you need smaller amounts of drug, then the single use also makes a lot of sense.
Okay. Well, I think we're going to wrap that up. If your question wasn't answered, Stuart will get back to you. So thank you, Stuart.
Thank you, Leah, and thank you, everyone, for joining today. We're happy to take any follow-ups. Please, just ping us on our website, and we'll get back to you.
Great. And the recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at our future BioProcess International Ask the Expert webcast. Look for those announcements in your inbox. Thanks again. Have a good day. Bye.